2 things left, and we are done with this chapter. I've been skipping around a little bit. This is going to be kind of an unconventional chapter. Your assessment will be unconventional as well. I don't think we're going to have like a normal test.
Is that like a good, like, organized the normal test, or is it like,
I'm going to really get you. No, it's good. It's fine. No, it's fine. It's just, it's time related and importance related. Like, I want you to know about this stuff, but I don't need you to like draw out how it works for me, because we need to get to the next stuff. So coming further into the present, you know, the stuff I taught you about how to clone a cat, that's kind of old tech. That's like 30, 40-year-old technology. Sounds really cutting edge. It's not. It's easier now. The question of why do you have to clone the cat to make a glow-in-the-dark cat? The answer being because we need to get that gene that makes you glow in the dark into every cell in the cat's body, right? So the first proposed solution to that, like how do you do this on a large scale with someone who's already an adult, is something called gene therapy. Basically, instead of picking one cell and sticking recombinant DNA in that one cell and getting a new gene into one cell, one tool that will do that for you, that will insert DNA into a cell for you, is viruses. Gene therapy basically involves designing a virus that you then infect an individual with. Now, normally harmful viruses, if you'll remember, we discussed this early in the year. Normal viruses stick their DNA in a host cell. The DNA codes for, do you remember what viral DNA usually codes for? Usually codes for more virus. Makes a bunch of virus parts, busts the cell open, makes you sick. What if, instead of injecting DNA that codes for more viruses, what if the DNA that it injected into host cells was helpful? What if you could cure a genetic disorder by having a virus insert a gene that cured that disorder?
So it's like in movies when they reprogram a robot to be on the good side,
It's very much like that,
like Terminator.
except tiny, tiny little robots. And these poor scientists, these poor brilliant scientists that made this breakthrough, because we've been talking about it for, 30 or 40 years. Finally, in 2012, they used it to cure genetic blindness. 3 different people had their congenital blindness cured by a helpful virus. Amazing. And completely overshadowed by the next breakthrough in genetic engineering. Just bad luck that this came right before CRISPR, which I bet you've heard of. Raise your hand if you've heard of CRISPR. Ooh. Cool. Because CRISPR, the breakthroughs with CRISPR have basically made this already obsolete, because CRISPR is so easy to use the program to basically edit DNA however you want. So here comes CRISPR. It's actually, the full name is CRISPR-Cas9, which is built very much on a molecule you know already, which is RNA. It's largely an RNA machine. Basically, scientists have known about CRISPR for a long time. It is basically the bacterial version of an immune system. It is how bacteria fight off viruses. It's a lot like an RNA polymerase. You know, the enzyme that runs along the DNA and pairs up RNA, except instead of pairing up RNA, what CRISPR does is it runs along bacterial DNA, looks for viral DNA that's been injected by bad viruses and fixes it. Okay, so it searches out, removes, and replaces viral DNA from bacterial DNA. So we've known about it, but it took until basically right around when gene therapy was starting to work for a couple of scientists named Jennifer Doudna and Emmanuel Charpentier to figure out how to mess with CRISPR, how to basically program it to work in any cell, not just bacterial cells, to work in even human cells. And to be able to mess with the recognition sequence to essentially replace or edit any gene in a human cell, turn any gene on or off. I mean, I don't want to exaggerate here, but it literally changed the entire game. Like, we can do anything we want now.
Is it like we got the blueprint for the body and we can just like take stuff?
Anything you want. Pretty much in your kitchen. Like, it's easy to use. The stuff in your bioethics paper about, you know, editing babies, genes, designer babies, the stuff about like wings or claws, you can totally do it. Whatever you want. And here's where we get to the part where, as soon as scientists realized what this was and what it could do, that's when the meetings really started up. Like the scientists from around the world going, "Oh crap! What's allowed and what's not? What are you allowed to do? What are you not allowed to do?" Raise your hand if you listen to podcasts. Good. I have one for you. Huh?
Podcast anymore. I lied.
Right. Too late. No, it's good. It's about CRISPR. It's interesting. I think you'll like it. I'll ruin it with questions, of course. But yeah. Good. That is chapter 15, or all we're going to do of it. All right, your assignment is twofold. Number one, you have the bioethics paper that is due 2:30 tomorrow. Is that what I said? Yeah, 2:30 tomorrow. It's due digitally. You have another digital assignment in Classroom that I'd like you to open up now. Oh, I had the wrong one.
Oh.
It looked like the same thing, but it's different. So the link you have in Classroom is to actually one of your textbooks virtual labs. It's back to DNA fingerprinting. I think it's a better lab than the other one. So you're going to give it a shot. You're going to be my guinea pigs. So the doc should be in there now. Before you really dig into the virtual activity, go back to the Google Classroom thing, and the doc that goes with it should be there now. At a certain point, you're going to have to copy and paste the image of your gel fingerprint thing into the doc, so don't miss that. Have that open. But I'm just going to give you the time to work on it. Please let me know any kinks or problems that you have. I know I went through part of it, and it can be a little delicate where you have to click. So if there's something where you feel like it's not working, just move your mouse around real kind of in small increments and see if you can get to the part where it'll successfully click.